Hello everyone, welcome back to Vigyan Marg. So today we are going to discuss a very important topic of biochemistry for first year MBBS student, that is blood glucose regulation. It is a very important topic in the aspect of viva and long answer, short answer questions. So we will be discussing in detail about the topic. Every doubt will be cleared in this video. So before going ahead, please make sure to subscribe the channel. Make sure you subscribe the channel to get the latest videos on different topics of MBBS and NEET counseling related videos. So join the Telegram channel. Join the Telegram channel to access this PDF and other PDFs before proceeding ahead. So let's get started. So first question come in this topic is, why do we need to regulate the blood glucose level? So see, most of our vital organs like brain need this glucose as its metabolic fuel. Not only brain but also our vital or other vital organs need this glucose as its metabolic fuel. What is metabolic fuel? The glucose is used in different metabolic pathways that is happening in these organs, especially like brain. In mostly all of the metabolic pathways we need glucose. This is the source of energy for our vital organs. So in this blood glucose regulation, a multiple number of hormones and huge number of metabolic pathways are uh, involved in this blood glucose regulation. So this is a finely tuned homeostatic mechanism occurring every day in our body. The most important and finely tuned homeostatic mechanism. Okay, so blood glucose blood glucose refers to the concentration of the glucose in blood. It is tightly regulated to ensure energy supply to the cells. Disruption can lead to serious metabolic disorders. If the supply of blood glucose is decreased or increased, there may be malfunction of the metabolic pathways and lead to metabolic disorders. So we need to regulate this blood glucose in a normal level, not hyper, not hypo, at a normal range. So at first we are going to see the sources of blood glucose. So what are the sources from where we get this glucose in our body? So at first, dietary intake. So through dietary intake we take carbohydrate, and this carbohydrate is broken down into glucose, and this glucose is provide us energy. Next is glycogenolysis. So glycogenolysis is the process in the process in which the glycogen stored in our muscle and the liver is converted to glucose. Glycogen is converted to glucose, and this glucose provides the energy. So you can see this mainly in liver. Why in liver? Why not in our muscles? So see, the glycogen stored in our muscle is cannot be converted to glucose directly. Why? Because a enzyme called glucose 6 phosphatase, which is absent in our muscle. That's why the muscle glycogen cannot be converted to glucose, and in the glycogenolysis the glycogen stored in the liver is converted to glucose and used as the source of energy in case of deficiency of glucose. Next is gluconeogenesis. Gluconeogenesis. In the gluconeogenesis, you can see the neo. This neo means something new from where glucose is synthesized or something other sources than glucose. That is formation of glucose from non-carbohydrate sources. That is non-carbohydrate sources like lactate, amino acids, and glycerols. Okay, so let's see. So the sources of glucose vary in different aspects like oil fed stage, early fasting, prolonged fasting, and uh, starvation, prolonged starvation. So in the oil fed stage, what is the oil fed stage? Within 1 to 4 hours of our food intake. This is the oil fed stage, 1 to 4 hours of our food intake. So 1 to 4 hours within our food intake is a fed stage. In this stage, the source of glucose is dietary glucose, which is the carbohydrate. This carbohydrate is broken down in pieces of glucose by the process of glycolysis, and it's provide the glucose. Next is the early fasting. So early fasting stage is 4 to 16 hours without food intake is the stage of early fasting. In this stage, our hepatic glycogen is converted to glucose to glycogenolysis. So I have already told you why the hepatic glycogen, not the muscle glycogen. Okay, in the previous slide. So next is the uh, so the stored glycogen is depleted within the stored glycogen is finished within 16 to 18 hours. So what will happen after this stage? We will provide this glucose. So next, the source of glucose is gluconeogenesis, that is in the fasting stage. What is the fasting stage? The time 16 to 48 hours without any food intake. Without any food intake is the stage of fasting. In this stage, the source of glucose is gluconeogenesis. I have already cleared what is gluconeogenesis in the previous slide, so no need to clarify here. Next is prolonged fasting. Prolonged fasting is also called starvation. So what is the definition of starvation? 2 to 5 days without food intake is a stage called starvation. In this stage, there is no direct source of glucose. So what is the source of glucose? Ketone body synthesis. Our stored fat is oxidized, which is called fatty acid oxidation, and is produced ketone bodies, which is the uh, source of energy. Not so direct source of glucose is ended here. Okay, still here gluconeogenesis, there is a direct source of glucose where you can our body can use glucose as a source of energy, but after that our body start fat oxidation and muscle proteolysis. By this our body uh, fulfill the energy requirement. So next and last stage is prolonged starvation. Prolonged starvation. So if you don't take food more than 5 days, 5 days without food intake is the prolonged starvation stage. In this stage, our muscle proteolysis means the structural protein of our muscle get proteolyzed and produce the energy. So what are the methods of estimation of blood glucose? So there are many methods you can see, like fasting blood glucose. So fasting blood glucose is a normal level of fasting blood glucose is 70 to 100 milligram per deciliter is the normal level of fasting blood glucose. So how we measure the fasting blood glucose? So in a patient who is fasting overnight, that is more than that is more than 8 hours. Who is fasting for more than 8 hours, we take the blood sample and we measure the fasting blood glucose. Next is postprandial blood glucose. So in this case, a patient who have taken food in last 2 hours, don't take any food for last 2 hours, and then we measure his blood glucose level. The normal level is less than equal to 140 milligram per deciliter. That is 2 hour duration. Okay, that is 8 hour duration. That is 2 hour duration. Okay, so in this stage, we see that how our body is reacting to glucose and managing the glucose level. So next is random blood glucose. Measure blood glucose at any time regardless of when the patient ate. Regardless the regardless when the patient ate last. Okay, this is random blood sugar. And there is another method called OGTT. What is OGTT? Oral glucose tolerance test. In this method, we measure how well your body process sugar after you drink a sugary drink. Okay, so patient is given a sugary drink, and in several time gap we measure the blood glucose and see what are the changes in his or her body and how his body is managing this increased amount of glucose. So there are some enzymatic method also, that is called GOD-POD method. So before going ahead, we will discuss GOD-POD method in less time and detail. So GOD-POD method is glucose oxidase and peroxidase. In this method, glucose is oxidized and it is oxidized by hydrogen peroxide, and it produces gluconic acid and hydrogen peroxide. This hydrogen peroxide convert this into a uh, pinkish, or you can say pink color dye, which by the concentration or the intensity of this dye we can estimate the blood glucose level, which is called glucose oxidase or peroxidase method. Next is hexokinase method. In this hexokinase method, glucose plus ATP with the help of hexokinase is converted to glucose 6 phosphate plus ADP. And next, glucose 6 phosphate is converted to, by glucose 6 phosphate dehydrogenase converted to 6 phosphogluconate, 6 phosphogluconate plus NADPH. And next, this NADPH is absorbed light at 340 nanometer, 340 nanometer, absorb light at 340 nanometer. The intensity of glucose tolerance is directly equal to absorbance of light. So absorbance is directly proportional to glucose tolerance. Okay, so let's see what are the normal levels of plasma glucose. Okay, so no, in the normal fasting, that is 70 to 99 milligram per deciliter, and postprandial less than 140 milligram per deciliter. Diabetic ranges in fasting stage is more than equal to 126 milligram per deciliter, and postprandial stage it is more than equal to 200 milligram per deciliter. So what are the importance of regulation of blood glucose? To prevent hyperglycemia, it is increase of blood glucose more than normal level, and hypoglycemia, decrease of blood glucose less than normal level. Ensure continuous supply of energy to brain. I have already told you about this, and muscle. Maintain metabolic balance, which is metabolic pathways. Okay. Stages of maintenance of blood glucose. So first is absorptive stage. Phase stage we will discuss in the detail after this. Okay. So let's take a look just, which is insulin, where fed stage, when the blood glucose is high, when you take food and the amount of glucose is enough and high in your body, so our body releases insulin to manage this high level of glucose. Next is post absorptive stage, which is fasting stage, where the blood glucose level decreases and our body releases glucagon, which rises the blood glucose by different mechanism, which we are going to see later. Glucagon rise. Starvation shift to fat derived ketone bodies, which we have already discussed. Okay, in the starvation state, in a prolonged starvation, brain adapts to use ketones, that is ketone bodies. Okay. So next is hormonal regulation. So there are lots of hormone regulating the blood glucose level, but the hormones do not directly interfere the uh, uh, that do not directly regulate the blood glucose by interfering directly. They interfere in this process by metabolic pathways. They do not interfere directly. They just interfere in metabolic pathways. By these metabolic pathways, the blood glucose is regulated. So these hormones plus metabolic pathways togetherly control the blood glucose regulation. Okay. So how does our body operates the blood glucose regulation to this by this metabolic pathways? So let's see. So there will be two scenarios. One is well fed state. In this well fed stage, what will happen? The amount of blood glucose is high. Blood glucose is high. So what will happen? It will inform the pancreas. So what pancreas, in the pancreas, it activates the beta cells of pancreas. So it inform the beta cell of pancreas. So the beta cell of pancreas will release insulin, and this insulin, how does the insulin regulate the blood glucose? The insulin will increase the glucose uptake. By which? By GLUT4, which is a glucose transporter present in the muscle and adipose tissue. Okay, in the mainly in the cells, but not in the liver. In the liver, there is GLUT2, which is the transporter in the liver. Okay, and by increasing glucose utilization. Okay, so next is fasting stage. So in the fasting stage, how our body regulating the glucose by these hormones. So in the fasting state, our body releases glucagon from the alpha cells of pancreas. What will release? Release glucagon, which is a hyperglycemic hormone, which increases the blood glucose and take it to the normalized level. Okay. So what will happen? This glucagon firstly decreases the glucose uptake. By insulin, it was increasing the glucose uptake by GLUT4, and by glucagon, it is decreasing the glucose uptake by GLUT4. Okay. Next, it increases the glucose release from where? By uh, gluco glycogenolysis and gluconeogenesis. Okay, glucose release, and next it increases glucose synthesis from non-carbohydrate sources and from glycogen. That's how it increases the blood glucose and take it to the normal level. So let's see how the insulin regulating the blood glucose. Okay, so in the well fed stage, the uh, insulin glucagon ratio is high. In the well-fed stage, insulin glucagon ratio is high. So in this stage, insulin when released from the beta cells of pancreas promotes glucose uptake. We have already discussed glycogenesis and lipogenesis. Like, what is lipogenesis? Lipogenesis is formation of fat from glucose. So when you take more amount of refined sugar, you are taking refined sugar unstoppably. So the extra refined sugar will be stored as fat. Okay, this is called lipogenesis. It will be relaxed in the fat. Inhibit gluconeogenesis and glycogenolysis, which is formation of glucose from non-carbohydrate sources and glycogen. Okay, so let's see in the detail how does the insulin regulate the glucose in the well fed stage. So in the well fed state, the increase of insulin glucagon ratio. Okay, so well fed stage means the blood glucose is high, so increase blood glucose level. So in this increased blood glucose level, our beta cells of pancreas is stimulated, and the insulin release. So what does insulin do? Insulin activation of various pathways by dephosphorylation of enzymes. That is happen when the insulin glucagon ratio is high. So insulin will be released, and what will do? Activation of various pathways by dephosphorylation of enzymes. When is the insulin glucagon ratio is high insulin glucagon ratio. Okay, so what will happen? The insulin will increase GLUT4. By increasing GLUT4, what happens? Increases glucose uptake by, first is heart, second is skeletal muscle, third is adipose tissue. It will also, in the other way, inhibit pathways utilizing inhibit pathways generating glucose, and other hand it will stimulate the pathway utilizing glucose, by which utilization of glucose is increased and the blood glucose get reduced. Utilizing glucose. Okay, so by this increase of glucose utilization. Okay, so what are the metabolic pathways and effect of insulin due to phosphorylation? So let's see in a brief. So what is happening here? Firstly, glycolysis is increased. Secondly, glycogen synthesis is increased. Thirdly, glycogenolysis, which is formation of glucose from other than carbohydrate sources, is decreased. You can now think why it is decreased. I've already discussed it. Fourth, glycogenolysis, which is formation of glucose from glycogen. Okay, gluconeogenesis, which is formation of glucose from other than carbohydrate sources. So gluconeogenesis is decreased, obviously. Fifthly, lipogenesis. So what is lipogenesis? Let's see what is lipogenesis, then we will say it will decrease or increase. So excess sugar you have taken through uh, diet, by which is refined sugar. Okay, you have taken more and more sugar you are going to take, and this will store as fat. So how the process happen in our body? So excess glucose, which is converted to pyruvate, and it increases pyruvate dehydrogenase and convert to acetyl coenzyme. Okay, this acetyl coenzyme is converted to fatty acid by glycerol. Okay, and this fatty acid stored as triacylglycerol. Now you can see the excess, where does the excess sugar goes? It's stored as fat in your body and relax there for many times. Okay, so this stored sugar or this fat will oxidize when you will be fasting for long days. This fat will degrade and produce energy by producing ketone bodies. Okay, so lipogenesis will increase. Okay, so next hormone is glucagon. So glucagon is secreted from alpha cells of pancreas. We've already seen that. Promotes glycogenolysis, formation of glucose from glycogen, gluconeogenesis, formation of glucose other than carbohydrate sources, lipolysis, formation of ketone bodies from fat. Opposes insulin action. What are the insulin actions? It does exactly its opposite. Okay, so let's see how this Glucagon act on the sugar and it's regulating the blood sugar. So this is the hyperglycemic hormone, means it increases the glucose level when the glucose level decreases from the normal level. Okay. So in the fasting stage decrease insulin glucagon ratio. So fasting stage, there's no dietary glucose, there's no source of dietary glucose. So what will happen? No dietary glucose. So our alpha cells of pancreas will release glucagon. Release glucagon. So what does glucagon do? Glucagon activation of various pathways by phosphorylation of enzyme, same as insulin. Okay, insulin activates different enzyme and glucagon activates different enzyme for different pathway. Insulin activates different enzyme for different pathway. That was for storing of glucose and utilization of glucose. That it is for utilization of stopping utilization of glucose in the cells and increasing its level in the blood and formation of glucose from other sources. Okay, so glucagon activation of various pathway by phosphorylation of enzymes. When it happens, when the insulin glucagon is ratio is low insulin ratio. So what will happen in this stage? Decrease activity of GLUT4. Decreased activity of GLUT4 is used for utilization of glucose in the cell, like skeletal muscle and our adipose tissue. So, if we decreases the activity of GLUT4, the utilization of glucose will decrease, and it will decrease the glucose uptake. Okay. On the other hand, it decreases the glucose utilization and increases the glucose generation. Okay. So let's see in brief what is happening in the fasting stage. What are the metabolic pathways which is increasing and decreasing? Okay. Very first, this chart is very important. You can mark it. In this chart you can clarify the overall topic. Okay. So glycolysis is decreasing, obviously. Second is glycogenolysis, which is in the early fasting stage, that is 4 to 16 hours, is increasing. Thirdly, gluconeogenesis is increasing, which is the fasting stage, that is 16 to 18 hours. Fourthly, lipolysis, formation of ketone bodies from stored triacylglycerol. Can you remember in the previous in regulation by insulin we have learned that how the excess sugar is stored as triacylglycerol. In this case, the excess stored triacylglycerol is converted to ketone bodies, which is the metabolic fuel. So that's the exact opposite of the thing happen in the insulin regulation by insulin. So lipolysis lipolysis prolonged fasting or starvation stage, we have already seen that. So stored triacylglycerol is converted to glycerol and fatty acids. This fatty acid gone through beta oxidation, okay, and form acetyl coenzyme. This acetyl coenzyme form ketone body, which is the metabolic fuel. Okay. So what is the hormone used in this case? The hormone used in the whole process is hormone sensitive lipase, which is also increased. So lipolysis is increased. Okay. Role of other hormones. So what is there is also other hormones which regulate the blood glucose, like cortisol promotes gluconeogenesis, epinephrine increases glycogenolysis, growth hormone reduces glucose uptake, promotes lipolysis, thyroid hormone. You can see these all hormones are hyperglycemic. So now you can think that there are many hormones which are glycemic. So what are the hormones which are hypoglycemic working against these all hormones? So there you can divide these hormones in two parts. One is hyperglycemic, one is hypoglycemic. Okay. So what are the hypoglycemic hormone? Glucagon, epinephrine, glucocorticoids, growth hormones, adrenocorticotropic hormone, and thyroxine. So you can think, if can you tell the what are the hypoglycemic hormone which is decreasing the blood glucose level? So you can hardly remember one name, the huge hormone, the huge monster which is fighting against all these hyperglycemic hormones. That is insulin. This is the only one hormone fighting against all these hormones and decreasing the blood glucose level. Can you think the power of this insulin hormone and the importance of this insulin hormone in our body? So next, the organs involved in maintenance of blood glucose. So there are many organs, you can see liver, pancreas. There are muscle which is regulating the blood glucose level by storing it or releasing it. There is adipose tissue which store glucose as fat. Brain constant glucose demand. Okay, so these are things that regulating also regulating blood glucose. From here, the liver and pancreas are the most important organs, or there are organs which are regulating blood glucose, and it plays a very important role in the regulation of blood glucose. Okay, so now see the role of liver in regulation of blood glucose. So this is the, here you can see this is the central organ in glucose homeostasis. Okay, the central organ in glucose homeostasis stores glycogen, performs gluconeogenesis, releases or stores glucose based on hormonal cues. Okay, so effect of organs upon release of insulin from the beta cells of pancreas due to increased blood glucose. So we'll see how our body organs like liver and the skeletal muscle and adipose tissue are controlling the blood glucose when the blood glucose is rise, that is in the well fed state. We are seeing this case in the well fed state. Okay, so blood glucose is high. So very first, let's see the in the what is happening in the liver. So you take glucose from dietary source. Okay, glucose. This glucose is converted to glucose 6 phosphate. Okay, this glucose 6 phosphate is converted to glycogen in the liver by glycogen synthesis. Okay, so if we take more and more glucose, this glucose 6 phosphate will convert it to pyruvate. This pyruvate will convert it to acetyl coenzyme by pyruvate dehydrogenase, convert to acetyl coenzyme. This acetyl coenzyme give will used in TCA cycle. So this TCA cycle happens in liver and glycogen will be stored in the liver, and this pathway from glucose, it is glycolysis, you already know. So this pathway glucose to acetyl coenzyme, it's common for all uh, where this process happen is common for all. Okay, so let's see what are the things that are increasing in the glucose. So in the liver, glut2 is increased. Okay, so by this, can you tell me what are the things that are increasing? Glycogen synthesis, obviously glycogen synthesis will be increased. Next is increased glycolysis, we already seen that. Glycolysis. Next thing, increases pyruvate dehydrogenase, and next thing is increased TCA cycle. Okay, these things that are increasing in the liver. So next is the fat, which is adipose tissue. So what is happening in the adipose tissue? The same pathway is same from glucose for adipose. For adipose, glucose is converted to acetyl coenzyme by the same pathway we have seen before, acetyl coenzyme. This acetyl coenzyme will form fatty acid. Fatty acid is converted to triacyl glycerol with the with combining with glycerol. This process is called lipogenesis. We have already seen it before. So you can see we are revising multiple times, same thing multiple times, so you can remember it very easily if you listen the things properly and with focus. So what are the things which is increasing in adipose tissue? So in the adipose tissue, GLUT4 is increasing. So increase in the glucose uptake, increase glycolysis, increase pyruvate dehydrogenase, increase, main thing is fatty acid synthesis, which is lipogenesis increased. So next is skeletal muscle. What is happening in the skeletal muscle? So in the skeletal muscle, the thing happening in the liver is same. Both the things is same. Same reaction written on liver. Same to same is happening for the skeletal muscle. Okay. So what are the things increasing in skeletal muscle? Obviously, GLUT4 is increasing. GLUT4 is increasing. So increase glucose uptake. Next, same glycolysis, increase pyruvate dehydrogenase, increase TCSIR, same as liver, increase glycogen synthesis. Okay, so these are the things happening in the skeletal muscle. So lastly, we'll see the uh, thing. So we have already discussed what is happening when the in oil phase stage, when the blood glucose level increases, and how the adipose tissue, liver, and skeletal muscle is handling the glucose. So in the case of decrease in blood glucose level, the opposite thing, the same and opposite thing will happen. Exactly opposite thing will happen. So we'll see it in the brief. So you can just relate the things. Don't try to mug up and just try to learn the things by revising, and don't mug up, don't confuse, don't get confused. Just relate the things, that the things happening in the insulin and the glucagon is directly opposite. Okay. So in the fasting stage, de- decrease blood glucose. So we'll draw three columns. Okay. So one is for adipose. You can relate it, just see it, you can relate. Next is skeletal muscle, and next is liver. So you can just see it in one slide, just compare the things. So what will happen in the adipose tissue? So in the insulin, in the case of insulin, uh, in the organs or the muscle in the adipose tissue, by insulin it was lipogenesis. So what will happen in this case of fasting or the decreased blood glucose? In the increased blood glucose there was lipogenesis. So in the decreased blood glucose there will be lipolysis, which is the formation of ketone bodies from the stored triacyl glycerol. So what will happen? Lipolysis. We have already seen the lipolysis reaction. So stored fat, stored triacylglycerol, which is releasing glycerol. So just opposite the reaction, nothing difficult. Fatty acid, beta oxidation, and it will give acetyl coenzyme, give ketone bond. Okay, see, same thing reaction, just opposite. In the skeletal muscle, glycogen. So in the skeletal muscle, what will happen? In the skeletal muscle, I have already told you in the very beginning, the glycogen stored in the skeletal muscle cannot be released directly in the blood because of the absence of an enzyme glucose 6 phosphatase. Because this enzyme is absent in skeletal muscle, the glycogen stored is converted to alanine, and this alanine is a gluconeogenic substrate, which is a non-carbohydrate source. So this non-carbohydrate stored alanine is goes to liver, and in the liver this alanine is converted to glucose and released into the blood. So there is no direct release of glucose from the skeletal muscle. This from the skeletal muscle, the alanine goes to liver, and the liver will convert this into glucose, and glucose will be released into blood. Okay, now we'll going to see this. How does this happens? Increases glycogenolysis will give glucose 6 phosphate. Next is glycolysis and give pyruvate. This pyruvate will convert into alanine, not into glucose or not into acetyl coenzyme, nothing. This convert into alanine, which is a gluconeogenic substrate. Okay, this alanine is going to liver as a non carbohydrate source. This will give glucose, and this glucose is going to release into blood. So another source of glucose in the liver is glycogen, which is stored, which will directly convert to glucose, and this glucose released into blood. So you can see the whole pathway here. You can see the whole pathway here, which is connected with the liver and skeletal muscle. Okay, this alanine is going to liver, and as a non-carbohydrate stored, this non-carbohydrate stored is gluconeogenic substrate, and gluconeogenesis happens here. Gluconeogenesis, gluconeogenesis is happening here, and the non-carbohydrate source alanine is converted to glucose and released into the blood. So you can see the complementary role of insulin and glucagon. Decrease blood glucose, glucagon increase blood glucose. Okay, togetherly maintain homeostasis in response to feeding and fasting. Negative feedback loop. How the uh, so when you eat glucose or eat food, so what will happen? The insulin will keep releasing, or it will be stopped at a level. Yes, by negative feedback. It happens by negative feedback. Okay, blood glucose level. See, blood glucose level is high. Blood glucose level is high. So it will release beta cells. It will give a feedback to beta cells and release insulin. This insulin liver removes glucose from blood and stores glycogen. So blood glucose level decreases and it's give a negative feedback to the pancreas and decreases the release of insulin. And the same, when the blood glucose is too low, pancreas releases glucagon. Okay, liver breaks down started glycogen and release glucose into the blood, and it will give a negative feedback to pancreas, and it will decrease the release of glucagon. Okay, so see, blood glucose level after eating, pancreas beta cells detect high glucose. Okay, insulin is secreted into the bloodstream. Okay, insulin acts on liver glycogenesis, muscle and fat. Glucose uptake increases by GLUT4. By glycogenesis, glucose is stored as glycogen. Okay, blood glucose levels is decreased. By blood glucose levels decrease, blood glucose level decreasing. So what will happen? Lower glucose reduces stimulation of insulin release. Reduces the stimulation of insulin release, and blood glucose returns to normal range. Return to normal range. So homeostasis is maintained. So let's see what is hypoglycemia. Hypoglycemia, that is when the blood glucose reduces reduces than the normal level of blood glucose, that is less than 70 milligram per deciliter. Okay, can lead to brain dysfunction or coma in severe cases. So low glucose levels is a very severe problem. In this case, there may be brain dysfunction or brain dead, or you can go into coma. Factor decrease in blood glucose, excess insulin, prolonged fasting, intense exercise, alcohol. Alcohol also can decrease the blood glucose. Insulin secreting tumors. So if there is any tumor in the pancreatic beta cells, there will be excess stimulation of the beta cells, and there it can result into the increased amount of insulin secretion, and there will be a very dangerous condition. Symptoms of hypoglycemia. So what is the symptoms of hypoglycemia? Sweating, palpitations, tremors, irritability, confusion, dizziness, loss of consciousness in severe cases. Next is hyperglycemia. So in the hyperglycemia occur when you can tell a patient that you are glycemic. You check the patient's blood glucose level. It comes out more than 140. More than 140, you can postprandial, which is postprandial blood glucose. I have already discussed what is postprandial blood glucose. So you can now see what is the importance to see the how we measure the blood glucose. It is very important to see how we measure the blood glucose. So if I directly say you the postprandial blood glucose, you will be confused on what is postprandial blood glucose. So this is about blood glucose after 2 hours of taking food. Okay, so I have already discussed it. So you have to know the methods by which we are measuring blood glucose. Okay, so associated with diabetes mellitus. So in the next lecture we will be seeing the types of diabetes is happening in our body. Okay, factor increasing blood glucose, insulin deficiency or resistance. So in another case of diabetes mellitus, there is insulin resistance. When our cells do not respond to insulin, or the receptor in our cells do not respond to insulin, so the insulins can't act on the glucose transporter 4, and they're thus decreasing the utilization of glucose. That is glucose resistance. Okay, so next is high carbohydrate intake. So if we take more amount of sugary drink, or more amount of refined sugar, the that thus increasing the blood glucose. Stress hormones, infections, sedentary lifestyle. Symptoms of hyperglycemia. So what are the symptoms of hyperglycemia? Polyuria, polydipsia, polyphagia. Okay, fatigue, blood vision, slow wound healing, frequent infections. So you can see there's increased thirst, frequent urination, extreme hunger, blurred vision, slow healing of cuts, fatigue. These are the symptoms of hyperglycemia. So in the summary, so you can read it by your own. So you can see glucose is the is a toxic substance we are taking, mm, is killing every day us, but we don't take it as a measure. Okay, it's killing every day silently many humans, but this is the major problem is diabetes nowadays. It's become a severe problem in the youngsters also. Okay, so you definitely check your glucose levels, and you may be pre-diabetic, post-diabetic, or diabetic. So that's it for today. In the next lecture we'll be seeing about the types of diabetes, and in the next lecture we will be completing all this topic in detail. So to watch more these types of lecture, make sure to subscribe the channel. Okay, to get the notification, press the bell icon, and definitely make sure to join the Telegram channel to access the PDF. Okay, so thanks a lot for watching. See you in the next lecture.